Here is our presentation on mitosis by Jerry Chen and Luke Richardson.
The first step of the cell cycle is interphase. The cell cycle is a series of events in which a cell grows, prepares for division, and divides in order to form two daughter cells.
So in interphase, the DNA replicates, and also in this stage, the DNA is loose and is also uncondensed. In interphase, the cell also grows in size and does normal cell activities.
Here's a picture of the cell with a nucleus and uncondensed DNA, and you can see as the cell grows in size throughout interphase. The second step of the cell cycle is prophase. In prophase, the nuclear membrane breaks down and DNA condenses into chromosomes. Chromosomes are thread-like structures in the nucleus. It carries genetic information that is already stored in the nucleus.
Chromosomes are also made of two chromatin, which are joined together by a centromere. And a chromatin is a DNA thread that consists of DNA tightly coiled around histones. And now in our diagram, you can see the former nuclear membrane, which is represented by the dashed lines, and also the chromosomes, which are represented by the X-shaped structures. So metaphase is the third stage in the cell cycle and also the second stage in mitosis. So in the metaphase stage, chromosomes meet at the equator, which is the middle of the cell.
One copy of the DNA is on each side, and these are called chromatids, as you can see in the picture, with the equator dividing them in the middle. The fourth step in the cell cycle and the third in mitosis is called anaphase. And in anaphase, the DNA separates by chromosomes, or centrioles, that are organelles that work together with spindle fibers that aid in cell division.
So the centrioles aid in cell division by pulling the chromosomes apart, as you can see in the diagram, so then it leaves the chromatids on opposite sides of the cell.
The last stage of mitosis is telophase, and mitosis is a part of eukaryotic cell division where the cell's nucleus divides.
In telophase, DNA that has spread out to the opposite sides replicate, and the new nuclear membrane forms around them, and also the two new nuclei form too.
As you can see in the diagram, the nuclear membrane reforms in this stage, and the DNA is ready to join with DNA from another cell.
Cytokinesis is the last stage of the cell cycle. In this stage, cytoplasm divides, providing two new identical cells. And in this form, the DNA is in the form of chromatin, which is loose and uncoiled DNA.
A quick review is the cell cycle is a series of events where a cell grows, prepares for division, and divides. Chromatids are one of the sister parts of a duplicated chromosome, and the cytoplasm is the fluid in the cell where chemical reactions take place.
Thank you for listening to our presentation on mitosis and the cell cycle.